So we're now going to go to the warm-up. Warm-up. Let me share my screen. And here we go. Again, as you can see, a lot of people are kind of solid, but then a number of people really kind of more lost. Again, these warm-up quizzes, I'll say once again, they are here kind of as little check-ins to make sure that you're keeping up. If you are taking them and realizing that you're missing a lot of stuff, that's that's an indication that you need to kind of adjust how you're approaching the class if you want to do well. Let's see. First one. All right, so this is Krebs cycle. We'll be talking about this in much more detail in another week or two. This is happening in the mitochondria as part of cellular respiration. But as you can see, it's just a bunch of molecules that are going to another molecule, to another molecule, to another molecule. This is a classic example of a metabolic pathway, right? That's what we were talking about. Most of the chemical reactions are going to happen in multiple steps. And what is the word we use for the molecules that are made along the way that are not our final stopping points?
Intermediates.
What are the intermediates? And then there's always going to be some kind of enzyme that is moving you from one intermediate to the next. This one, I was kind of shocked. What describe the function of enzymes and what are their defining properties? Not every, a lot of people were kind of lost on this. What is at the core, what an enzyme does?
Speed up the
Enzymes speed up chemical reactions.
Yeah, they speed up chemical reactions. There's a lot of people who knew they have to do with chemical reactions, but didn't really have a solid sense of, they are making something that normally wouldn't go very fast in the normal conditions, all of a sudden run very quickly. And run basically very quickly in the conditions available in the body. And then what's another defining characteristic of an enzyme besides speeding up reactions?
They aren't consumed in the process.
Exactly, they're not consumed in the process. So they come in, they make this enzyme substrate complex. We talked about how the substrate binds to the binding site, or bind to the active site, I should say. And then the reaction happens, and then the enzyme is now detached and free to do it all again. And again, what kind of biomolecules are most enzymes made from?
They're made of proteins.
They're made of proteins, which we'll talk more about today. And that shape of that active site is critical for them functioning properly. This one, again, 71% got this right, but a third of the class got this wrong. If you're one of the third of the class that doesn't really get what a mole is, you got to figure that out pretty soon, because moles are kind of our core way of specifying amounts and concentrations and things like that, right? 1 mole, what is a mole of an element? What does it mean by definition to have a mole of carbon or a mole of sodium or a mole of helium?
6.02 times 10 to the 23rd molecules or atoms.
Yeah, exactly. And if we're talking about elements, then it's about atoms. So it would be 6 times 10 to the 23rd atoms of helium, which will float up into the sky, right? You put those in a balloon and it floats. or it could be 6 times 10 to the 23rd atoms of lead. That's definitely not going to float, right? So they definitely don't weigh the same. You know, the same number of atoms of one element can have a very different mass than the same number of atoms of another element. So really make sure that you kind of disentangle that if those are confusing to you. Back here, so this is good. Only one person missed this one. Hydrogen ions increase, pH goes down. Again, this can get confusing because things are going up and down. pH is going up, alkalinity is going down, hydrogen ions are going up, pH is going down, hydrogen ions go down, pH is going up. So make sure that you are clear about this. This one, a number of people missed. And so we really need to make sure we talk about this and it's really clear for you, because this is going to be a concept that's going to be also going to prevalent throughout the entire semester. This idea of osmolarity versus molarity and what happens to something when you make a solution. So if I'm maybe I'll just get out of this and share my let's say this is sodium chloride. Let's say it's 2 molar sodium chloride, which means 2 moles NaCl per liter. What's the osmolarity going to be?
4.
It's going to be 4, right? Whatever the molarity is, it's just going to be 2 times to get to the osmolarity, because every NaCl becomes an individual Na plus and Cl minus. So every single NaCl becomes 2 particles. So whatever the concentration and molarity this is, it's going to be double that as the osmolarity. Does that make sense? What if it was calcium chloride instead?
6.
Exactly, because this is going to be 3 times. Every calcium chloride becomes a calcium and 2 chlorides. So there's 3 particles for every one of these. So that would be a 3 times. What about this glucose? But if I had 2 molar glucose, what would it be in osmolarity?
2 molar, I mean 2 osmolarity.
Yeah, exactly. Because every glucose stays intact. This becomes just a glucose floating around. So it's just going to be a 1-to-1 in terms of molecules of glucose and particles in solution. Right, so make sure this makes sense, because this is going to be at the core of lots of stuff in our lecture and in our lab. A lot of the upcoming labs are going to be looking at osmolarity, and you'll have to be calculating osmolarity of electrolyte solutions versus non-electrolyte solutions. Again, if it's covalently bonded, the osmolarity is just going to be equivalent to the molarity in terms of number. If it's ionically bonded, then you have to figure out how many ions does it dissociate into. So any questions about that? Okie dokie. All right, so now we are gonna continue. We're gonna I hope we're gonna be done with our chemistry review today, because we actually do have to start talking about the kind of physiology.
Do we have Quiz on Tuesday.
Yes.
And it'll just be like chem review.
It will be on everything up through what we talk about today.
Okay.
It will be administered through Proctorio, which is part of the reason why we need to make sure everybody has Proctorio working before we're done with our class today. I want to make sure all the bugs are worked out. So when we take, when the stakes are real, when we're taking an actual quiz, that you have the system working. This quiz is going to be 10 points. So this is worth a little more than one of these warm-ups are, again, just kind of check-ins. This is worth more points, takes longer. Again, we're doing through the Proctorio system. And again, instead of just mainly being focused on the stuff just from the class before, this is going to be focused on everything from the first day of class up to today. And it will be after lecture. We'll do lecture first, take a break, and then do the quiz. So the quiz will be kind of like in the beginning of lab time.
And that is going to include lab or just lecture?
Just lecture.
Just lecture, okay.
Just lecture. Although, our lab has been covering some lecture material as well, right? So the chemistry review lab is basically, covering a lot of the core concepts from lecture. All right. Now, so there are a few main classes of molecules that make up most of the stuff that we're going to be looking at. And it's worth spending a little bit of time looking at the detail of how these molecules are put together and their properties and the implications of that. So we're going to be looking at carbohydrates. We'll be looking at lipids. We'll be looking at proteins. And we're looking at nucleic acids. Right. Yeah, this is also the class like when you're looking at your little nutrition on the side of your like food, talks about carbs. These are so let's look at each of these in turn and start with carbohydrates. So carbohydrates, these are always made out of carbon, hydrogen, oxygen, and always in the ratio of 1 to 2 to 1. It's going to be different. Like when we get to lipids, you also have the same kind of molecule, the same kind of atoms, but they're put together in very different ratios. So you have these, big hydrocarbon chains coming off of these things. But if you're a carbohydrate, it's always going to be 1 to 2 to 1. Part of their name comes from this. Like when people knew that they had this ratio, they like, oh, it looks kind of like, H2O and carbon, hydro, water is carbon, hydro, water, carbo, carbon. So they're not really carbons and water, but the ratio, that's where the name comes from. It gets its name because it's like an H2O and carbon in terms of the ratio. There are 3 main subcategories in the carbohydrates. Monosaccharides, disaccharides. And polysaccharides. Monosaccharides are often called simple sugars. So monosaccharides, this includes like your pal and mine for cellular respiration, like glucose. C6H12O6. Right? C6H12O6, 1 to 2 to 1 ratio carbon, hydrogen, oxygen. This is an example of what we call a hexose sugar. Because it has 6 carbons. There are other hexose sugars that have the exact same formula, but are assembled in slightly different ways that are also hexose sugars, like fructose, galactose. They all have the same chemical formula, but they're assembled in slightly different ways. And there's actually different versions of glucose even. There are also pentose sugars. Hexose sugar with 6. You can also have which would be, whoops, C5. H, what would the H have to be?
10.
H10. O5. What's a important pentose sugar that is at the, it's like the backbone of all of the genetic material in your body?
Ribose?
Yeah, ribose. This would be like ribose, deoxyribose. Those are pentose sugars, 5 carbon sugars. So these are the monosaccharides. When you put them in water, they take on this ring formation. Like when I am drawing sugars, I'm usually going to draw them as like a little ring. Like the other carbons actually coming off. There's 5 of the carbons in the hexose sugars are part of the ring, but then there's an oxygen, and then there's other things hanging off. So, you know, this class, you are not ever going to have to, on your own, draw the detailed bonding of the carbons and hydrogens and oxygens in a glucose, but you should know that this is glucose. You should know that it basically looks something like that. You know, I'll show you some actual pictures of it later. I mean, you actually saw a picture of it when you had to draw it when you were doing your chemistry review lab. So monosaccharides. Disaccharides, from what it sounds like, that's just the prefix for 2. This is when you take 2 of the simple sugars and you connect them together. And depending which ones are connected together, you get different sugars. If you have a glucose and a fructose, this is going to be sucrose. This is your classic transport sugar in plants, right? This is CNH pure cane sugar from Hawaii. This is the or from beets or whatever. You know, the reason why sucrose is so important is that's one of the main sugars we end up eating, because that's what the plants make, and then we eat the plants, or we'll grind them up and extract the sugar from them at least. Other disaccharides are going to be, if you have a glucose just connected to another glucose, that's maltose. If you have glucose connected to a galactose, that's milk sugar, that's lactose. Right, so, and again, if you are going to ultimately take these and use them in cellular respiration, you're going to have to break them down into the simple sugars, into the monosaccharides. You'll have to break that bond and separate them. And so you can feed them into the metabolic pathways that are designed for, the monosaccharides. Right. And what if you don't have the enzyme that breaks apart glucose and galactose?
Isn't it
You're
like
lactose intolerant.
Then you're going to be lactose intolerant. Your body is not going to be able to break this apart. And it's just going to make it through your digestive system into your large intestine, where there's going to be bacteria that are more than happy to break it apart and make you gut all gassy and weird. Right, and this is kind of genetic. A lot of adults don't have the enzyme that metabolizes lactose. While we're at it, just some of the terminology that people use for either assembling or disassembling things. Let's say I have a sugar, and again, I'm going to just make it kind of this ring, and hanging off of one of the carbons is an hydroxyl and OH. Here's another sugar. There's another OH hanging off. So here's two monosaccharides that I want to assemble into a disaccharide. What happens is, again, I should. We had two OHs. The link between the two ends up just being an oxygen, which means two H's and an O disappeared. So we lost a water. So sometimes people call this like dehydration synthesis, because you are synthesizing, you're building this thing and you lose a water, dehydrating, it's evaporating, so to speak. If you're going to do the opposite, if you want to break this thing apart, then we have to add a water in. So to make two separate things, we need to bring the water in and add it to the system. So the word that we often use for this kind of reaction is hydrolysis. Hydro, water, lysis, to cut apart. You've probably heard of hydrolysis, right? It just means cutting apart, and it's taking a water molecule to do it. So hydrolysis is just kind of breaking apart. All right, so disaccharides are going to be important, particularly because they're the main sugars you're getting from your diet. Polysaccharides. Polysaccharides are a bunch of simple sugars connected together. The ones that we're going to care about are a bunch of glucoses connected together. What do you call it when you just have a whole bunch of glucoses all connected together in big long chains?
Is starch.
That is starch. Right? Starch, this is the main storage carbohydrate that plants use, right? You are a carrot or a potato or some other plant, and you're storing away your energy for a rainy day. You're making lots of starch. And then some human comes and rips you out of the ground and chews you up and swallows you. So that's where we get our a lot of our energy from as well. Or, if you're a wheat plant and you're putting lots of starch into the seeds so your new little embryonic babies can grow and become strong plants, but then some human comes along and rips you out of the ground and grinds up your little seeds and makes flour and bakes it into bread. So starch, this is where we get a lot of our glucose, and it's easy to just break each of these bonds and free up the glucoses because we have the enzyme to do that. However, plants also have another polysaccharide that's also made out of glucose put together in big long chains. I'm going to just try to make it look different by making it have a different looking bond in between. So here a human has the enzyme necessary to break starch apart into glucoses and use those glucoses. Another polysaccharide used by plants more usually as a structural element. It makes up the cell walls of their cells. It's the main thing that makes up wood. Is what?
Cellulose.
Yes, cellulose. So cellulose, we do not have the enzyme to break the bonds between there. Right? There's obviously a lot of energy in wood. Right? You put it on, you light it on fire and you make a nice campfire and you warm yourself. So there's lots of energy, you can free it up in the appropriate situation. But if you just eat a bunch of wood, you're going to poop out a bunch of splinters, right? Your digestive system does not have the enzymes to free up those glucoses and actually use them. There are animals that can eat, lots of cellulose, like, ungulates. Like if you're a cow or a deer or something, what you have is you chew it, you swallow it, but then you actually have these kind of symbiotic bacteria that are sitting inside your stomachs, and they break down the bonds of the cellulose and free up the glucose, and then you can regurgitate and swallow chew your cud and swallow it, and then extract the nutrients from that processed cellulose that the bacteria have already fermented. The bacteria in your intestines can break stuff down as well. That's, the roughage, like when you eat lots of beans, that, why do you toot when you eat the magical fruit, so to speak? So like where you've got the little bacteria that are breaking down cellulose that you could not break down and absorb, but they're breaking it down and making gas in the process. Cellulose is what we call the fiber or roughage in our diet. And it actually is important. Even though you don't get nutrients from it, it gives bulk to your feces to keep the feces moving along, keeps it hydrated so it doesn't get stuck and you don't get constipated or impacted bowel, even worse or something. So having cellulose or fiber to kind of bulk out your feces and give it something that the walls of your colon can actually push against is actually important. The other polysaccharide, there's only one more polysaccharide that I want to mention. So starch. This is what plants use as their storage carbohydrate. Animals have something really similar. So kind of regular plant starch is a bunch of glucose kind of strung up in these big long chains. What do animals do that's similar?
Glycogen? No?
Yeah, no, exactly. You are correct. It tends to be more branching. Often people just call it animal starch. So your body stores lots of glycogen. So if it, your liver has lots of glycogen. So if your blood sugar starts dropping, it can just turn on an enzyme and start nibbling away and freeing up these little sugars into your bloodstream and keep your blood sugar up. Your, particularly your fast twitch muscle fibers are full of little glycogen granules to make ATP really fast through glycolysis if you need some quick energy in your muscles. So glycogen is used in your body to store energy and release it quickly as glucoses. It's not nearly as efficient. You can store like over twice as much energy in the same weight of fat. So you're not going to use glycogen as your main storage in your body, but it's a way to quickly be able to release glucose into the body so it can be used immediately. So any questions about carbohydrates?
Is glycogen also broken up with hydrolysis?
Yeah, the hydrolysis is the kind of generic term. Yeah, because remember, each of these is just two sugars connected together with a little oxygen. So yeah, it would take hydrolysis. In general, that word ends up getting used a lot. A lot of these equation or reactions that we're going to see are similar like this. Starch. Let's move on. Let's do proteins next. So proteins. Proteins are used, obviously, these are going to be enzymes. These are also going to be important structural elements, right? This is making up your cartilage and connective tissue and stuff like that. Proteins are also going to be important kind of in the membrane, doing all sorts of functions. Controlling what goes in and out of cells and detecting things as receptors and things like that. We're going to be seeing enzymes and structural proteins, membrane proteins, other kinds of proteins. So it's important to understand what a protein is, what its properties are. When we get into like what it means for a neurotransmitter to bind a membrane receptor, you actually have to understand the detailed structure of a protein in order to make it make sense. What does it even mean to bind a receptor? And then why does that actually cause something to happen in the cell? So we are going to dive down a little more about how proteins are put together. What is the building block of all proteins?
Amino acids.
Amino acids. So we should start here with what is an amino acid. And it's basically amino, blah, blah, blah. Amine group, it's a nitrogen. The acid is going to be a carboxyl. And so this is the acid, the amino, but then there is this side chain. And the side chain, in our body, there are 20 in our proteins. There are more than 20 amino acids, and the other amino acids play different kinds of roles, but there are 20 main amino acids that make up all of our proteins. And this side chain can have very different chemical properties. can be polar, nonpolar. It might make, positive or negative ionic bonds. It might want to accept an electron or give up an electron. So it has there's many possible properties. So depending on which amino acid you're looking at, you're going to have very different properties on that little part of the the protein. So this is going to be critical when we start looking at what does it mean to bind to an enzyme or bind to a receptor, because different parts of the protein like to do different kinds of chemical bonds. All right, so then we're going to start attaching these amino acids together, basically end to end with what we call peptide bonds. So we're going to call the first order structure of the protein again, we call these peptide bonds. What determines which amino acid gets connected to which next amino acid?
The RNA transcript.
Yeah, exactly, right? Originally, there's the code in the DNA, but then it gets transcribed into the messenger RNA, which then brings it up to the ribosome. And at the ribosome, the transfer RNA is bringing amino acids up based on the codon, right? I know you all in Bio 110 all go through this, so this should feel familiar. So the DNA code is basically which amino acid to hook up in which order. So this is what we call the primary structure of the protein, is just the order of amino acids that are connected. There are often a lot, thousands, tens of thousands of amino acids. In fact, if it's less than 100 amino acids, we usually just call it a peptide instead of a protein. There's a lot of important peptides we'll talk about. There's a lot of signaling molecules, things like insulin. Insulin is just 37 amino acids connected together to be considered a peptide. Most of the proteins that are making up, like enzymes and things like that, they're like tens of thousands of amino acids connected together in these really big, complicated molecules. Okay, so first, this primary structure is just the order of the amino acids. But now we're going to get more and more complicated. We're going to go next to the secondary structure. There are two different types of secondary structures that are common. In one kind, the chain, the snake of amino acids, starts coiling around and making these spirals. This is called an alpha helix. Basically what happens is, oh, I did not draw this so well. This is like 1, 2, 3, 4. Every third amino acid kind of hydrogen bonds to the one above it in the helix, so it stabilizes it, right? So it's a stable structure that forms as amino acids hydrogen bond to the one like right above it in the spiral. So alpha helices Another possible thing that we find are what are called beta pleated sheets, in which case you have kind of the hydrogen bonding is more across kind of walls of these. So this snake, we have a snake of amino acids, but it has two main secondary structures it can fall into, either these alpha helices or these beta pleated sheets. So that's the secondary structure. This is just, I realize if you don't know, this is the Greek letter alpha. This is the Greek letter beta. Then you get even more elaborate, complicated folding happening, because this is, you have kind of hydrophobic and hydrophilic things going on. Remember I said some amino acids are nonpolar, some are polar, some are going to be positive polar or negative polar, some are going to want to go out towards water, some want to fold away from water. So this thing, we get what's called the tertiary structure. So it gets to be this big, and some proteins are more globular, some are more fibrous, but the basic idea here is this, you get this more complicated folding. And if you look, some of it are these helices, some are these beta pleated sheets, but then it's folded in a bigger context as well. Some of this just happens naturally due to the chemical attractions of the different parts of the molecule, although there are kind of little chaperones and stuff. There are actually other molecules in the cell that will actually help fold the proteins properly. And at this point, many proteins are functional at this point. What I should mention, there's also more. There's also like disulfide bonds. There's like places where you get like sulfurs going across and locking in to stabilize the shape and stuff. So it's more than just hydrophobic, hydrophilic, but basically you get this big complicated shape. Many proteins are functional at this point. Here, let me go to the fourth, the tertiary structure, and then I'll come back here in a moment. There is a optional tertiary. Two or more, sorry, quaternary, right? I spazzed there. So two or more of these tertiary third order subunits to make the functional protein. We'll see a lot of this. Like one of the classics is hemoglobin in your red blood cells. Hemoglobin has 4 globin subunits to make a functional protein. Myosin, the contractile protein in your muscle, has these heavy chains and light chains. Antibodies have heavy chains and light chains. So there's quite a few important proteins that we'll be seeing that are made up of 2 or more subunits to make the functional protein. And then on top of that, sometimes they need other things. When we start we'll talk about cofactors, coenzymes, things like that. There are certain things that need additional things in addition to just the protein to be functional. Hemoglobin isn't going to work actually without iron in there, stuff like that. All right, let's go back then. So this 3rd degree tertiary structure. So here it makes more sense now, like why pH is going to denature a protein, right? If I start putting a whole bunch more H pluses out here, there's all this extra charge, and that charge, certain things are going to want to get pulled to it or pulled away from it, and it's all going to bend differently, right? So you can denature this, with, pH, extremes of pH. If you're changing the charge surrounding this thing, it's going to mess with it. You can denature it with temperature, right? Because temperature is just that motion. So if I start making it more and more agitated and it's starting to vibrate more and more, it's like, oh crap, it's now all tangled in on itself. We're never going to untangle it. Right? That's what happens when you boil an egg white, right? Egg white, albumin, is a protein, and it's this kind of white goopy, it's kind of clear, goopy, looks like snot kind of. And then you boil it, you heat it up, and all of a sudden it becomes this big solid block. And it's because you've entangled all of those amino acid chains, and irrevocably, you're never gonna like pull them apart into individual little albumins again. Right, and you can obviously denature with pH, right? Ceviche is using the lime juice with your fish, and it's basically looks just as if you cooked it at higher heat, right? Because it's basically denaturing the proteins. You can denature it mechanically as well. Just for completeness, like when we're talking about eggs, what's the other way to turn albumin into solid white aside from cooking it?
To whip it.
Separating it.
Yeah, just whip it, right? Whip it in a meringue. You're basically taking all of these proteins, and they're physically getting moved enough to where they're starting to get entangled with each other and stuck together. So again, this denaturing is important to remember, because denaturing, the shape gets messed up. And if the shape gets messed up, it's not going to be able to interact in the way that it used to. Particularly, we're going to see that with enzymes or with receptors or things like that. Let me see if I have a picture here that'll be helpful. So this is just, just reviewing monosaccharides. And again, in water, they make this kind of ring structure. You do not have to know the details. You are not going to be required to draw any of these or anything, but you should know what they are. You should know that any pentose sugar is, C5H10, and that any hexose sugar would be C6H12O6. But you do not need to know the subtle differences between a fructose and a glucose. You know, I couldn't do that without looking at a paper, so don't worry about that. This is not a chemistry class. This is showing disaccharides, again, connecting up, you know, a glucose and a fructose become a sucrose, two glucose is a maltose, a galactose and a glucose is a lactose. So again, just what I was drawing, but much prettier. And again, you can see the 6 carbons are all there, but they're not all in the ring. Like the 6th carbon is up here, right up here. There's an oxygen in the ring. So just, this is showing glycogen. This is just a bunch of glucoses connected together. This is showing starch here. Starch, it's one continuous chain instead of more of a branching structure. Again, cellulose looks a lot like starch, except the bond between the glucoses is slightly different, and mammals do not have the enzyme that can break it apart. Any mammal that wants to digest cellulose has to have a symbiotic pal, like usually bacteria or protists or something that work with them. Termites even have to have symbiotic protists and stuff to digest the wood that they're eating. So not even a termite has it's always these little microbes that can break down the cellulose directly. Okay, amino acids. Here's the amino acids. And again, it's got a carboxyl, an amino, and then this R is a side chain. You connect them together as big chain with peptide bonds. Primary structure is just which one is connected to what. Like I said, usually they're called proteins when they're more than 100 amino acids, and peptides if they're less, if they're shorter. Secondary structure, you've got alpha helices and beta pleated sheets. This tertiary structure here, you can see you've got this thing with the beta pleated sheets and the alpha helices in this big crazy structure here. Quaternary structure, this is like a hemoglobin showing 4 of these things connected, and this actually has the little heme groups as well in there because it actually needs this iron containing thing to be functional as well. So, but you should know the primary structure, secondary structure, tertiary structure, and quaternary structure. So any questions about those? Okay. So now let's do fats. So fats are very similar in, well, actually, I'm gonna come back to this in 1 second. Let me give you a overview and then we'll go to some specifics. So lipids, they are also lots of carbons, hydrogens, and oxygens, but way more carbons and hydrogens. They have these big, long hydrocarbon tails. So the ratios are way different compared to a carbohydrate. As a rule, one of the biggest things you need to remember in this class is lipids are usually hydrophobic. I'm going to say are hydrophobic. So in general, when we talk about something being a lipid, it means it's going to have trouble dissolving in water. So when we talk about steroid hormones, which are lipids, they are not going to just be able to dissolve in your plasma. They are going to need special carriers to actually move around through your bloodstream. When we are absorbing fats from your diet in your intestines, you can't just bring them in like you'd bring in an amino acid or a glucose, because they don't dissolve in water, and they're just going to be a much more complicated system to absorb fats from your diet and bring it into your body. So it's important to know that lipids are hydrophobic, and that's going to mean we need different ways of dealing with them, different ways of carrying them around through our bloodstream, all that kind of stuff. Now I can introduce fatty acids. So let me introduce a normal, when we think of fats, usually we're talking about triglycerides. So a triglyceride is basically a glycerol. Glycerol is just some alcohol. And to the glycerol, you attach 3 fatty acids. Each fatty acid is basically a little carboxyl with a big long hydrocarbon chain. This is, I'm doing kind of some generic thing. I don't know which one I'm drawing here even. But basically what you're going to have is this can bind to this. And the fat is going to basically be a glycerol with these 3 fatty acids hanging on here, which are basically hydrocarbon chains. So this is a classic fat. A fat glycerol kind of group here, and then these these are hydrocarbon chains hanging off. So does that make sense? What we need to talk, and then I can show you if I share this again. You know, these are showing these fatty acids, right, with the little carboxyl that's attaching to the glycerol, and these big long fatty acid hydrocarbon chains coming off. If you look at each of these, if you look at this top one, every carbon is got a single bond to the next carbon. This is what we would call a saturated fatty acid, because it has got as many hydrogens as possible with that kind of carbon backbone. If you have a single bond between every carbon, that lets you maximize how many hydrogens you can attach along the sides. It's called saturated fatty acid because it's saturated with hydrogens. It's got the maximum possible numbers of hydrogens that you can slap on there. This monounsaturated fatty acid here, it has this double bond between the carbons. At that point of unsaturation, you're only able to have a single hydrogen there rather than one on either side. So it's not as saturated as the other one, because it's got a few less on that big hydrocarbon chain. We have a polyunsaturated fatty acid here with lots of points of unsaturation. At each of these points of unsaturation, you have fewer hydrogens than you would have compared to here. So this is even more unsaturated than our saturated fatty acid. So does that make sense when we say saturated versus unsaturated fatty acid? Yes? So then what I should show you then, let's do this. What's our time? Let me just finish this thought here. So if I have a bunch of saturated fatty acids, so that's, I guess, the thing I need to mention. If I have a fatty acid and it's saturated, the tail tends to be straight. Whenever you have a double bond between the carbons, you get a bend in the molecule. So unsaturated right? So that point of unsaturation, it kinks. So if I have a triglyceride that's just made out of saturated fats, the hydrocarbon tails come down straight. If I have one that's made with unsaturated fats, the tails tend to kink. This leads to, I think, a pretty intuitive property of saturated versus unsaturated fats. You know, these obviously are going to be able to just stack on top of each other more easily. These saturated fats, these tend to be solid at room temperature. This would be lard or butter or something like that. If you heat it up and get the molecules jiggling, it'll turn to liquid. But if it's room temperature, it tends to be solid. Unsaturated fats, on the other hand, these tend to be liquid at room temperature. Most of the fats in plants are unsaturated, like corn or safflower or any of those. What's another word for these unsaturated fats that are liquid at room temperature?
Would it be oil?
Yeah, exactly. We also call that an oil. Monounsaturated, like olive oil, is monounsaturated. So olive oil is liquid at room temperature, but it doesn't take much to get it to solidify. You put it in the fridge, it turns like, solid. You've probably experienced that. Whereas if you took corn oil and put it in the fridge, it stays liquid because it's polyunsaturated. So it's got more of this kink in its tail versus, olive oil, where it's a little kinked, but not as kinked as a polyunsaturated. Let me look at the time. We might as well mention, just before we say goodbye to this, let's just mention the idea of hydrogenation. So unsaturated fats, one of the thing is they're oil, they're liquid. Also, they tend to be a little less stable. They'll break down, go rancid more quickly than saturated fats. People had the cool idea, actually in health studies, saturated fats tend to be more correlated with cardiovascular disease. If you're eating a lot of saturated fats, you're more likely to get cardiovascular disease versus eating a lot of unsaturated fats in your diet. So back, at least, when I was growing up, people would eat margarine. Margarine was this idea like, well, if unsaturated fats are healthier, corn oil is healthier than eating lard, let's just make corn oil look like butter. And hydrogenation just means let's go in and slam on a bunch of hydrogens and straighten out the tails so it will be solid at room temperature, and will have better staying power. For peanut butter, it's great because if you have natural peanut butter, the oil separates and you have to stir it every time you want to make a PB&J sandwich. Whereas if you open up your bottle of Skippy, it's like naturally creamy, ready to go. Because, you know, the oils have been hydrogenated, have been turned into a version that's solid at room temp. The problem, the stuff people found out a few years ago was in this process of hydrogenating the fats, that they were creating what are called trans fats. So right in, we have cis. We'll be seeing this a bunch. When you have, if I have 4 atoms here, I can have the same 4 atoms hooked up in the same order, but I can bend them one way or bend them another way. This would be cis, this would be trans. Trans would mean these two are across from each other. Cis, they're on the same side. So I can have the same chemical structure, but bent in different ways. So what was happening in this hydrogenation process, people were creating fats that had bends in them that were not naturally occurring types of fats. So the body saw these things and they did not have the enzymes to process them, because these trans fats that were created in this hydrogenation process were these weird things that are not naturally occurring. And it turns out that they were just as bad or worse for you than just eating butter. So it was very ironic. You know, now they've come up with ways to kind of get around this and avoid making these bad trans fats, but you've probably heard of them. And if you were wondering, if you didn't know what they were and wondering what are trans fats, it's just when you chemically alter the oils to try to kind of solidify them, and you make these versions that your body doesn't like and are not so good for you.
So are they not good for you because they make you gain weight, or they cause like digestive problems, or both? Or
I think they've been correlated with cardiovascular disease, inflammation, things like that. I'm not as familiar with all of that literature, but I definitely know that the original thought that like margarine was going to be way better for you than eating butter ended up being wrong.
They're a big component in the inflammatory response.
Yeah, so that makes sense. And we'll talk about this when we get to the cardiovascular system, but the like atherosclerosis, like, you know, the hardening of the arteries, that actually, you know, people think about it in terms of like, oh, the fats stick into the inside of your vessels, but that's not what's happening. It's actually an inflammatory response that's depositing lipids within the walls of the vessels. So yeah, all that cardiovascular disease are these inflammatory responses that are creating these depositions in the walls of arteries and all. All right. I think
Quick question for you.
uh-huh.
You said a triglyceride is a glycerol with, is it 3 fatty chain acids hanging off of it? Is that the correct nomenclature?
Yeah, it's a glycerol connected to 3 fatty acids.
Okay, thank you. Sorry about the dog.
Oh, that's all right. Lipids continued. All right, so we talked about triglycerides. Those are like the fats and the oils. There are other lipids that are important. Obviously, fats are super important for storage, for other things, for diet. Cholesterol is another lipid. It's got these interlocking rings. I can show you a picture of it. I can share screen. Again, you do not need to know exactly what the structure of cholesterol is. You need to know, though, that it is a lipid. And hence, if it's a lipid, it's hydrophobic. You know, and like all your steroid hormones are based on cholesterol. Cholesterol is also going to be an important part of your cell membranes. So again, you don't need to know the actual structure of it, but you do need to know that cholesterol and all of its relatives belong to lipids. Icosanoids, another word that describes a class of lipid molecules that a lot of signaling molecules in the body are based on. So again, if you see icosanoid, then you know it's a lipid. Again, don't worry about what exactly does an icosanoid look like. You should just know if you hear that word that it belongs in the lipid family. Phospholipids, on the other hand, phospholipids we need to talk about in some more detail here. So if you think about triglyceride, well, that's a horrible picture of a triglyceride. Let me get out of this. And if you think about a triglyceride as that glycerol with the 3 fatty acids with those big fatty acid chains going down, this is your classic fat. But you can also, you had those 3 hydroxyls that you were binding the 3 fatty acids to on the glycerol. You can also do another version of this where you only put 2 fatty acids on it. And on the 3rd binding site, you put this phosphorus containing group that's actually polar. So we are now going to talk about phospholipids. The basic idea here is you've got that glycerol with these hydrocarbon tails. So these are my little, I'm going to say non-polar hydrophobic tails, which is your classic, what you expect hydrophobic, you expect the lipid to be. But then you stick on this phosphorus containing thing that's polar. So this is going to be up here, this polar head. Only not, yeah, I'm writing kind of sloppy. Hydrophilic. It loves water. All right, so here we've got something that is not purely hydrophobic. Here we have something, half of it is hydrophobic, half of it is hydrophilic. The word people use is amphipathic, meaning it's got part of it that likes to be nonpolar interactions, parts of it likes to have polar interactions. You know, this is what detergents look like. You know, this is what your cell membranes, this should not look new to you. This is the core thing that makes up your cell membranes. So, and these will self-assemble. So if you take phospholipids and you just put them in water and you shake them up, you don't have to do anything. You just step back and all the hydrophobic tails are going to try to find each other to get away from the waters. All the polar heads are going to want to go out towards where there's water, and they assemble in what, you know, we call the classic phospholipid bilayer. Right, the tails go towards each other, so we create this kind of hydrophobic zone. And then where the water is, you know, all the H2O, we have the polar interaction. So the heads want to be towards the water The tails all want to be away from the water, so this self-assembles into what we call the phospholipid bilayer. Right, notice that there's no like particular bonding holding it together. It's sticking together because of the hydrophilic interactions, which also means that it's kind of fluid. So these things can slip and slide past each other. It's not like one phospholipid is glued to the next one. So what I will do now is, let's see, I'm going to show you another picture from the PowerPoint, actually the PowerPoint from the next chapter. It's going to be, here we go. I'm going to share screen. So this is kind of showing how phospholipids are assembling what? Wait, hold on, something went wrong here. I don't want this one. There we go. So here's this phospholipid stylized with a polar head and non-polar fatty acid hydrophobic tails. They can make a bilayer like this. This is more what your cells do. They make like basically a thing that has water in the middle. Where's this? I want a nice picture of the cell membrane. It's got to have a good picture. Oh, well, hold on. I apologize for this. I am going to this is better. So this is kind of showing how each of these pink circles is the polar head. All the little aqua squiggles are the hydrophobic tails. And you can see they just make, it's not just 2 dimensions, it's 3 dimensions. And this is what your cell membranes are. And you can also see that there's other stuff in there as well. There's all these membrane proteins that are embedded in there. So this is your classic fluid mosaic model of a cell membrane. Let me see if I got some other nice pictures of it as well. Yeah, this is a good picture of it as well. You know, you can see the little hydrophobic tails going down into this place in between. The polar heads are towards the water, either outside of the cell or into the cytoplasm. And then there's all these proteins that are stuck kind of in the middle of it. And think about each of these phospholipids. In fact, let me maybe get my, I'm going to get my annotator thing here. Draw. If I look at any one in particular, let me give it a color. Go on, draw. And I need a brighter color. If I take one of these little puppies, it's going to stay in the membrane, but it doesn't care if it's next to this one. This can easily move over, and a little while later, it might have drifted over here. And a while later, maybe it's drifted over here, and now it's sitting here. So that's what when I say fluid, these things are going to stay in the membrane, but they can shift and slide with respect to each other. And these proteins, this is supposed to be a membrane protein. This is a channel. It shows it's one of the ways things are going to get in and out. What is going to keep this in place? This is not glued in here either. How are we going to make sure that the protein that is in the membrane stays where it's supposed to be here?
Anchor it to something inside or outside the cell.
So it's, you could do that, but that is not, and in fact, and that wouldn't really make sure that it stays right in the middle of the membrane. So we will have things where we anchor onto the membrane to, like, for the cytoskeleton and things, but this is kind of a deeper question of why doesn't it just like pop out like a watermelon seed out one side or the other side?
Its structure in the side groups kind of poking out, like it stays inside the membrane because there's hydrophobic portions and then hydrophilic portions on the outside.
Exactly. So did everybody hear that? So you're going to remember we talked about a protein is made of amino acids, and some amino acids have polar side groups and some have non-polar side groups. These are constructed so the part that is going to be in this hydrophobic zone is going to be hydrophobic side groups. So all of the part of this protein is going to have non-polar. The top and the bottom, these are going to be polar. So if you try to like move it, if you try to push it one way, this side's going to say, no, I want to be towards the water. If you try to push it the other way, this side's going to say, no, I want to be towards the water. So this thing is going to stay in the membrane based on the polar, non-polar qualities of the protein. So does that make sense? And this can also shift. It's also fluid. It can also move around. I'm going to write this. I'm using a mouse. This is going to be very sloppy, but I'm going to write it the best I can. Oh, I'm doing okay here. Fluid mosaic model. Of the membrane. Fluid, because things are shifting and slipping and sliding with respect to each other. Mosaic, because it's not just one kind of thing. It's lots of phospholipids with embedded proteins, and not just embedded proteins, but also a lot of cholesterol stuck in between in the hydrophobic tails. So cholesterol is an important part of the cell membrane. The more cholesterol you shove between these hydrophobic tails, the less fluid it gets. So there are going to be different membranes with different properties, some that are more or less fluid, and the amount of cholesterol in there can make a difference. All right, the last thing I want to mention while we're here is understanding the nature of the membrane is going to allow you to think about permeability of the membrane. What can move in and out through the membrane with help or without help? So let's talk about membrane permeability. And again, let's remember the membrane is this phospholipid bilayer. Let's say this is outside the cell. This is inside the cell. This is my cell membrane. And again, this is H2O inside and outside. So let's talk about membrane permeability. There's something and it wants to, either it can go in or, something else tries and is rejected and can't make it. What's going to determine whether or not something can move across the membrane freely or whether it is going to be stuck on one side or the other?
Is polarity?
Big part is going to be polarity. So will polar molecules be able to cross the membrane?
Yes.
So if you think about it, let's say I am a sugar molecule, glucose. I'm coming up here and I'm polar, right? I'm starting to get into this hydrophobic region where there is no charge of any sort. And back here, it's like, but there's all the waters that want to do this hydrogen bonding. It's like, I'm not going there. It's no fun in the going into the membrane. It's like, I'm going to stay back here where there's all the stuff doing hydrogen bonding, pulling on me, and I'm getting attracted to. So a polar molecule, when it gets up there, it's not going to go and drift in and across. So polar molecules do not cross. Polar molecules are not going to go across because they're going to be attracted to the hydrogen bonding of the water. And that's going to hold them on one side or the other. And they're not going to have any motivation to drift into this hydrophobic. Remember, this is all hydrophobic in the region, hydrophobic tails. So there is an exception though, right? Because otherwise osmosis wouldn't be an issue for our class. So water, we talked about water tends to move in and out of cells because otherwise we wouldn't care about osmolarity. So it turns out really small polar molecules, people think about it kind of like a bullet or something. It's like, if I have a really small polar molecule and it's going really fast and it's like starting, it might be like, oh, whoa, wait a second, I'm more attracted here. But before it even knows it, it's on the other side. Kind of like, so this idea, polar molecules do not cross. Not permeable. Let me write this out prettier. Except if very small. For instance, water. Right, so H2O actually does cross. There's going to be a few cases where we have membranes that are particularly impermeable to water, even where you shove lots of cholesterol and stuff in there. But for the most part, you can assume water crosses the membrane. Otherwise, osmosis wouldn't be a theme of our next 4 months. Even though water is polar, it's so dinky. By the time it gets into the membrane, it forgets, it's on the other side before it even realizes it rather would stay with the other water. Another example that we're going to see in our lab next week, urea. Urea is a small polar molecule. It's going to be involved in metabolism of proteins and stuff, but it is small enough that it can actually get into the membrane and cross. It's not nearly as permeable as water, but it's still permeable enough that it makes a difference. It's going to affect our results, for instance, when we look at our lab in another week. So membrane permeability. Membrane, not permeable to polar molecules except if very small. So this kind of begs the question. We keep talking about ions in water, right? We talked about, sodium and chlorine. Could they cross the membrane? So let me make this a little prettier here. So let's say I have a sodium. Can a sodium cross the membrane without help, do you think? Sodium ion.
No,
Why?
because it's not small enough.
I mean, at the first, how big? It's a single atom. It's like as small as you can get, at least at first first glance, right? Remember we said small polar things, right? This is small, like water is 3 atoms and it's small enough to get across. This is a single atom, right? So in terms of size, it's small. But you are right, it doesn't cross. But it's not because in and of itself it's small. It's because of what happens, because it's so highly charged that it pulls the water to it. In this very rigid right. When we talked about a little H2O being polar and getting through, it's small, but it's charge imbalance. It's not pure charge. The sodium, on the other hand, is pure charge, and the waters all lock around it. It's basically like a snowball. It's basically ice, where you have this highly structured water forming around the sodium ion. They call it like the sphere of hydration or hydration shell. This whole thing actually acts as like a big snowball. So effectively, that sodium is really big because it is moving along with all of the water molecules that are being pulled really tightly on it. So again, it's the best way to think about it really is like kind of like a snowball. So even though the sodium itself is small, the snowball is big. And the snowball forms because the sodium or any ion is so strongly charged that it pulls the water into this highly regulated kind of crystal formation around it. So ions are going to need help. We're going to have all sorts of special channels that let ions cross membranes, but ions do not cross on their own. Does that make sense? All right. So the last thing we haven't talked about, what about nonpolar things? What would a nonpolar molecule do in this situation here? Assuming it could get, assuming somebody dropped it off at the membrane, right? It's going to have trouble floating around in the first place. But assuming it's made it to the membrane, Is it going to be able to cross or not cross?
Once it crosses, wouldn't it be stuck?
Right. So, but let's stick with the first step is it can cross because if it's nonpolar, it's totally at home here in the hydrophobic tail region. But yeah, Lindsay's right. Usually you're gonna have to get it there. Usually there'll be some carrier that brings it up and then it can go across and something else that picks it up and takes it on its way. But in terms of getting across the membrane, it doesn't need any special channel or anything. If it makes it to the membrane, it can just drift across to the other side of the membrane without any help. Whereas polar molecules, we're going to need, if I want to get an ion across, I'm going to have a special membrane protein that acts as a tunnel to let it get through. Whereas a steroid hormone, if it gets up to the membrane, it just ends up on the other side without any help. Does that make sense? So it's important that you understand and kind of as you start get a good feel for this membrane permeability. Without help, we're going to see lots of membrane proteins that help things cross the membrane. That's a whole other story we'll go into next week. But if you just have a phospholipid bilayer, so yes, for nonpolar or very small polar. No, most polar. So hopefully this, so yeah, if this is going to be why we're going to have all sorts of channels and carriers and things, because sugars and ions and amino acids and stuff, those are all larger polar molecules that we want to bring into the cell. So we need help. So any questions about that? We'll be talking lots more about cell membranes, way more about cell membranes, days on cell membranes, because that's kind of where the boundary between the outside world and the inside world is. And that's where cells sense their environment, how they communicate with each other. We'll spend a lot of time with the membrane, but first step is just kind of getting a sense of just the physical properties of the membrane here. Phospholipids with embedded proteins all drifting around with these kind of core permeability features. Okie dokie.